Primary mesenchymal stem cells create tissue mimetic structures in bioblocks. This video captures the cells growing on the opposite side from which they are seated, aka the bottom of the bioblock. This shows them organizing around the center pore and infiltrating throughout the block, suggesting we have cells on the top, the bottom, and inside the block. When compared to images captured on the top of the block, the cell behavior and organization is different, showing adaptation and that they are taking advantage of the topology of the growth surface. Primary mesenchymal stem cells, human adipose derived, have been cultured for 27 days without a subculture in Ronack's half X block. The stains are Hoeschst in blue to show the nuclei, Mitotrack in red to show the mitochondria, and actin in green to show the cytoskeleton and actin filaments. Primary mesenchymal stem cells create tissue mimetic structures in bioblocks, imaged in 3D on a Nikon Confocal microscope.